mice, rats, and a range of mammals will develop type 2 diabetes if you put them on a high-fat diet. And I think if you wanted a bit of evidence that saturated fats were bad for you, I think actually the animals give you the best evidence, better than any of the epidemiology data, because it's fairly good evidence from the epidemiology data, but it's only an association. Here we can actually do intervention studies and put animals on diets that will actually develop, they'll actually go on to develop type 2 diabetes, and we can watch them through that. The mice were originally thought to be resistant to developing cardiovascular disease and also type 2 diabetes until we looked at their diet. So most mouse studies will describe that they're on a regular chow diet for a mouse. If you look at what a regular chow diet is, it's basically like muesli. So it's relatively high in carbohydrates, relatively low in saturated fats, and as a result, mice don't develop diseases like cardiovascular disease in quite the same way that humans do. Having said that, if you then switch them onto a high-fat diet, you can very readily then induce atherosclerotic plaque, so they get cardiovascular disease, and they'll also develop type 2 diabetes relatively rapidly when you sort of move them on there. And to put these high-fat diets in there, they're often termed Western diets or the cafeteria diet. So they are meant to model what you get from fast food in terms of these animal studies. So you can generate type 2 diabetes sort of quite quickly from saturated fat. The other thing you can do are human intervention studies. If you've ever been interested in being a human intervention study, this is a plug from my sponsors at MRC Human Nutrition Research. So we do have a number of human intervention studies that are going on at the moment. So one of my PhD students is looking at what happens when we convert glucose into fat. I'll talk about that in a moment. He takes people and feeds them with large amounts of pasta, and then we see them how much they produce fat. But we have a range of different sort of intervention studies that go on, looking at how we can change diet for a short period of time and look at how that might affect certain aspects of disease processes. The problem is, it's not ethical to do it for longer. So the sort of Super Size Me, if you've seen the film, I'm pretty certain I would not get ethical approval to do that in Cambridge. You just wouldn't get it past the ethics board in terms of doing it. Probably wouldn't for mice or rats, actually, in terms of what you did. But we can look at sort of how things like increased saturated fats have been associated with changes in a range of outcomes, which are thought to be associated with disease processes. So you can look at some of the early stages of cardiovascular disease and the early stages of type 2 diabetes, change somebody's diet and see how that interacts with those processes. And there's fairly good evidence that saturated fat contributes towards insulin resistance and then hence type 2 diabetes. Problem with doing these types of studies is the effects are much smaller, so we get away with less in human intervention studies compared to animal studies, so we can take animals sort of on more extreme diets and get more conclusive data. So what I've hopefully sort of convinced you, there's fairly compelling evidence that saturated fats are bad for you. The question is though, what's going on with sugars? Why are we hearing so much about sugary foods are so bad? So the first thing to say is that we've known that type 2 diabetes is a disease of glucose since its first description. So if we go back, I forgot to put the dates on here, but I think these Egyptian and Indian doctors were talking about 2000 BC, describing honey urine disease, noting that ants will be attracted to the urine of people with type 1 diabetes, as the sugar that's present in there. And surprisingly, sort of 400 BC in India, they first made the discrimination between type 1 diabetes and type 2 diabetes. In Europe, we were a bit behind the time, but type 2 diabetes was known as the coachman's disease. I don't know if you're familiar with this, but the idea was that in order to find out whether somebody had diabetes, it required somebody to taste their urine. So the doctor's not going to do that, but his coachman would try it. So it's known as the coachman's disease. So I'm going to avoid my beer now. A little caveat though with this is that type 2 diabetes is also a disease both of sugar deregulation, but of also fat deregulation in terms of the disease processes. And probably if ants were interested in fat more and we could measure it more easily and we could taste it in the Coachman disease, we'd also be describing type 2 diabetes as a disease associated with fat metabolism as well as it being associated with sugar metabolism. So there is a question about, you know, sort of what's the causative thing? Is it fat or is it the sugar? Having said that, going back to the thing that in some ways that it's easier to look at these things in animal studies than it is in human studies. If you put mice on a high sucrose diet, they'll develop type 2 diabetes. So sucrose consists of glucose and fructose. There is a story out there that fructose might be particularly bad for us compared to glucose. There's quite a hot debate sort of in the nutritional field about whether fructose is particularly bad. The good news for us at the moment, we don't have high fructose drinks. You do in the US because of the corn lobby, but most of our sugar comes in the form of glucose or sucrose, but not fructose. There's also increasing evidence from epidemiology studies, but I've got to introduce a metabolic pathway for you first of all, and I apologise. This is what my undergraduates hate, and problem with glucose is you can't store large amounts of glucose. We've got a bit of glycogen that's found across the body, and glycogen is sort of what we use in that fight or flight response to sort of get away from a bear if a bear's coming to attack. But you can't have too much of that glycogen, and the reason for that is that glucose itself is water soluble. So it's a large amount of water that's around it, and the glycogen itself is not packed very tightly. Again, you know, if we've got to get away from a bear, we want a really open structure so we can get at that glucose and we can run away from the bear. Anyway, all that's saying is that it's not a great energy store long term in terms of metabolism. It's much better to actually convert this glucose into saturated fat and use that as a long-term energy store. The energy that we store in fat is much more efficiently put into that fat than the glycogen. So we have a process called de novo lipogenesis, so which sounds scarier than it is. I'm not going to go through all of this, but basically we go from glucose or sugar coming in through your diet, and if you're in the well-fed state, you can then convert that into fats through de novo lipogenesis down here, and then ultimately store it as triglycerides. So we can store it in our fat depots across the body, and it's much more efficiently sort of stored in terms of there. The nice thing about storing fat is that it's lipophilic, so it doesn't require any water associated with it, and it also has less osmotic potential. So the problem with glucose is that glucose drags water into cells and it causes cells to split, whereas if we can store it as fat, it has a lower osmotic potential and it doesn't cause the cells to lyse. It also explains why diets work right at the start of diets, and they don't work as you're going further on in diets. So if you're about to diet, the first thing that you're actually going to lose are probably your glycogen reserves. So your glycogen reserves are going to decrease, and they weigh quite a bit. It's the glycogen and the water that you're losing during that stage. Now as you continue to diet, your next port of call is the fats. Now we've just said that the fats are much more efficient in terms of that energy storage. So what many people when they're dieting sort of see is the pounds roll off first of all, and that's the glycogen and the water going, and then it's much more gradual, which is when the fat is going. Problem is that when that fat is going, it's probably doing the best thing for you, whereas glycogen itself is probably sort of largely beneficial, and certainly the water associated with it. So if you are dieting, what I would say is don't give up when you've hit that level, and if you could have a little sensor to tell you sort of how much fat you were losing, people would probably be much happier with dieting and see the beneficial effects sort of during that process. So that's why most diets work at the start, and they don't sort of work as you sort of go on. They are working, it's just much more difficult to get rid of them at that stage. So yeah, back to the biochemistry sort of 101. What I want you to remember is that via this process we can convert glucose into fatty acids, and we make 3 different fatty acids that contain 14 carbons, 16 carbons and 18 carbons. They're called myristate, palmitate and stearate, and they can be made from glucose. So I'm going to tell you about a study called EPIC. So this is a study which is partly being conducted here in Cambridge. EPIC is a large-scale epidemiology study for looking at cancer risk that's been set up to look across Europe. It's got 500,000 people from different countries across Europe involved in this, very similar to Finland, and we're looking at sort of risk of people developing breast cancer, colon cancer, and another cancer that I've forgotten. But there's 3 different cancers that are associated with this. Huge amount of investment into this. So we're involved in a study called Interact, which is piggybacked a component associated with type 2 diabetes and looking at relative risk of type 2 diabetes. We developed various robotics in Cambridge to measure the fatty acids that are present in people's blood. And the reason why we've done that is that we know the fatty acids that are developed in people's blood are associated with their diet. And we also know that some of those fatty acids are associated with developing relative risk of type 2 diabetes. So it's probably easier to sort of show you some of the results, and apologies for the people at the back. But this is relative risk across Europe of developing type 2 diabetes if you have high concentrations of 3 fatty acids, myristate, palmitate, and stearate. So these are the 3 fatty acids that I told you about. This is the relative risk over here, roughly about 1.5, so it's an increased relative risk of developing type 2 diabetes across all these different countries over here. Now the nice thing about EPIC is we've collected all the dietary data, what people are eating. So you can also look at what those fatty acids are most associated with. And again, you might not be able to sort of make it out sort of at the back, but in terms of what is most associated with this is we've got potato consumption, soft drink consumption, carbohydrate, I can't read it now. Margarine consumption, thank you, and alcohol consumption. So alcohol will make fatty acids. So if you have excess alcohol, you'll convert them into saturated fatty acids. Carbohydrates are very high in sugars, and potatoes are quite starchy, again are high in sugars. So these fatty acids, which are thought to be associated with increased risk of developing type 2 diabetes, are almost certainly sort of coming from sugars that are present in there, and we're making these fatty acids actually inside the body. So this is our big worry at the moment in terms of why sugars might be predisposing people to developing type 2 diabetes. Kind of frightening the margarine one for me, because my mum had switched me from butter to margarine, and the message actually is that butter really isn't that bad for you, at least in terms of EPIC and the diseases that we've looked at so far. So it's these saturated fatty acids that are present there. So what's going on in terms of our diet? So to speculate what's happened here is that we've had this public health message, which has been very successful, about cutting down saturated fats. Saturated fats, though, did taste nice in our food. So the food industry has now replaced those saturated fats with carbohydrates. And it's the carbohydrates which are having a knock-on effect. We've all got a sweet tooth, and those carbohydrates are now making fats inside our body, which are contributing to the relative risk of type 2 diabetes. And at the moment we're looking at experiments to try and sort of understand that sort of better mechanistically. So I'm aware that I'm overrunning on time at the moment, but so the one worry is that we're making these fats in terms of the liver. We're getting these liver droplets. So this is what fatty liver looks like. It's actually what it looks like in a rat, but this is 15 days of fatty liver induced in a rat. You get these large lipid droplets, and we end up with effectively sort of type 2 diabetes of the liver, insulin resistance in the liver, and that people are going on to develop type 2 diabetes. Slightly frighteningly, this can be in people that have quite moderate BMIs as well, particularly if you like beer, as it contributes towards the palmitate and the stearate. So the message is too much sugar or fat is a bad thing. Sugar may not be the bad guy because of changes in our sugar may now be the bad guy because of changes in our diet. So I think this is down to the fact that we've cut out a large amount of saturated fat in our diet. We've now got diets that are high in sugars. So also more than the first law of thermodynamics, so there's also things that will contribute towards this in particular, but both fats and sugar produce intermediates that are affecting our health. And then there's a couple of sort of weirder things that I sort of wanted to finish if I'm not sort of overrunning on time. The first thing is that actually it also makes a difference about when you eat. So you're probably all aware that we have a natural body clock inside our body. You probably think it's in the brain, and there is part of the brain which controls the circadian rhythm. But actually every cell inside your body probably is pre-programmed to understand, to have a circadian rhythm and a 24-hour clock. If you knock the clock out in mice in their fat cells, they'll actually develop obesity. And this is one of the reasons why we think that shift workers become predisposed to developing obesity and type 2 diabetes. So it makes a big difference about when you're eating the food. I have an Italian colleague who's a clinician, and he's saying that the Italians, because they've shifted from eating large lunches into large dinners, are now developing a thing called pasta belly. And this seems to be a major contributor to type 2 diabetes. So we might be able to treat type 2 diabetes just by moving forward dinner, or if you work from where I'm from, tea, to around about sort of 5 p.m, 6 p.m. That will have beneficial effects. There's help at hand, so dieting works. You'll also hear about gastric bypass surgery, so this seems to be increasingly being used. We don't quite, well, we understand sort of how it works sort of long term. The amazing thing is that gastric bypass surgery works within inside 24 hours. People have had reversal of type 2 diabetes symptoms inside 24 hours. We don't understand sort of how that works, but it's sort of a rather sort of extreme version of this. And then another possibility in terms of how we can treat type 2 diabetes is we can actually use our fat cells to burn some of the fat that's present inside our cells. So I don't know if you're aware of it, but on your neck you have some brown adipose tissue. So most of our fat is white adipose tissue. So that's where we store our fat. Brown adipose tissue can actually be used for keeping us warm. It contains things called mitochondria in there, which are used to burn fat and generate sort of heat energy as part of that process. Babies use it to keep warm, and if they don't have enough brown adipose tissue, it's thought to be sort of one of the reasons why they develop things like cot death. Adults weren't thought to have brown adipose tissue in any significant amounts until somebody started imaging young ladies from Newcastle. There's no young ladies from Newcastle, are there? and I'm told young ladies from Newcastle don't wear very much when they go out. And these young ladies then began to increase their expression of brown adipose tissue across their shoulders, and it just lit up on the imaging. And they're burning off their calories sort of in terms of their shoulders to generate heat sort of as part of this process. So I think I'm just going to sort of finish on the brown adipose tissue at this stage. So is that all right?
Thank you very much.